So this is going to be a short talk, so no worry. This is a collaborative work with CSUN and Ravi Abrol there, and NCBI, Jiao Wang and his team. And it might look like we took that as a small challenge. I don't know, for those who know CABAT numbering in immunoglobulins, the CABAT numbering gives you numbers for a VH domain, but other numbers even for a VL domain. And then you have others that worked on that with all, gave their name, Chotia and Martin and IMGT and so forth, always on antibodies. Now, what we were interested in is to understand the folding of the Ig domain across the whole proteome, all of them. And for us at NCI, we work a lot. Many of our targets are things like CD22, CD33, CD19. They're all, not all, but many are made of Ig domains. And then we target them with antibodies or scFv or nanobodies on top of cell surface receptors or with chimeric antigen receptors. So I was interested in developing something that would allow us to study any Ig domain. And that's how we got started. So the whole immune system can be looked at with a reduced number of folds, and in particular the Ig domain. But not just the immune system, the nervous system, the vascular system, the muscular system with titin, for example, oscurin. So all made of Ig domain. And Ig domains tend not just to be by themselves, they are forming chains. And you'll see later that if you study the whole proteome, the numbers of Ig domains can vary between 1 and 24, but much more for titin on the cell surface of proteins. So to us, it looked very important to look at Ig domains. And these are very well known for a certain number of them. The variable domain, the constant domain, C1 and C2, the intermediate IgI domain, and they all vary with some strands. And there are actually much more than just these 4 types of variants. If you take SCOPE, many of you know SCOPE or CAS or ECOD. These domains are categorized, let's say in CAS, as 330 superfamilies. That's quite a bit. SCOPE is what we used most, and they came up with more of a geometrical way of representing that with, let's say, 56 Ig domain, or Ig-like domain, or what we call Ig extended domain. So the first thing is to look at what's common and what's different in Ig domains. And I'm not talking about the loops, okay? That we all know they are variable, especially for antibodies. But the interesting part about Ig domain, apart from antibodies, is that they interact with themselves. They have a self-recognition, Ig, for most of them. And you can look at the parts that are invariable and the parts that are variable in an Ig fold. And actually, you can see that most of the places where you have variations are at the N or C terminus. So before the A strand, when you use the standard letters for representing strands, or the C terminus. But you also have in between the variations that you know most from variable to constant domain in the middle. And then you have other variants like in lamine and so forth. So we have to understand that this is a unique fold where even the topology changes. Most folds anywhere have a standard topology that hasn't changed. But here we have topology changing. So that's a paradox. And you can understand the paradox by supposing that the insertions are actually also forming strands that can extend the sheets. And that gives a tremendous flexibility to the fold to be able to create new interactions and have tremendous diversity. Now, the problem is that you all worked with PDB files and they have numbers, and the sequence numbers change even for very close proteins. And so we followed more the approach of the Ballesteros-Weinstein numbering in GPCRs, where they actually zoom in on anchors in the particular secondary structure elements, these anchors being the most conserved residues in terms of sequence. But with the IG, the variation is such that you cannot do that. So we focused on defining anchors structurally. So these anchors will have the number 50, and then the strand number will be the two numbers before. So for example, the anchor of the first strand, or A, will be 1550, meaning that you would have 50 for the most conserved residue structurally, 5 for its reference number, and 1 for the strand number. So ethically, what you're going to end up with that is a suite of numbers that are still manageable as to represent the sequence for any IG domains. So I should probably speed up. So now the strength of the numbering is that it will allow you to look at not just one IG, but interactions between IGs in terms of these numbers. So you're going to have positions that will be systematically the same in different superfolds that would be quaternary structures. So here you have CD8 dimer versus one antibody variable VHVL domains, and you can see that some interactions are systematically conserved. Now, we've done a scan of 250 or more antibodies from the COVID saga, and you can see that some of these interactions are systematically conserved, including those that are particularly symmetric. And that's true for the constant domain in the Fabs as well. And what's more interesting is when you move to other IG domains like lamine, for example, you start adding strands, and these strands may actually create different types of interactions. For example, arrestin, before I speak about arrestin, lamine would actually create a dimer using the strand before A, what we call A minus now, which also has a number, reference number. And there you can see the interactome between two lamines. Arrestin does the same. Now, the fact that it has an extra strand allows, instead of having a head to tail kind of arrangement, it has a head to head arrangement, because the A minus strand will actually reverse the direction of the second IG, giving you a super domain, if you want, that will bind GPCRs and so forth. Now you have many of these extended IG domains in transcription factors. You find IG domains everywhere. So with our numbering, we developed also an algorithm that can scan the whole proteome. And what we find is that among the 20,000 proteins produced by genes, it's not 2% that contain IG domains, the way they're defined, but more like 5 to 11%, depending on how you actually match the reference protein. Now, there is even more diversity because exon skipping can exist. And this is a very big deal for resistance to CAR T-cell therapy, for example. So you have to know which IG domain you need to target with your antibodies. Now, All that is done in ICN3D, our software that has an algorithm to actually recognize IG domains, each strand, and number all of them. Therefore, you can actually study any type of interactions among any type of IG chains, and you can compare them and so forth. And then once you've done that, you can actually exchange this information through a URL that will just give the person you send that URL to the software, the analysis, the visualization. And so if you want to know more, because there is a lot more with 10% of the human genome, you can look at the paper that we just put on, the preprint we just put on bioRxiv. And with that, I will take one question if there is one. Thank you.